Thank you so much for the introduction. Um, okay. Um, so yes, I want to start by introducing you to GRIN disorder, which is one of the many thousands of new rare disorders that's being identified now with the increasing prevalence of whole exome sequencing and genetic diagnosis. So people who have GRIN disorder were previously diagnosed with having developmental delay, intellectual disability, autism, epilepsy. And when their genome is sequenced, it's discovered that the cause of all of these really debilitating symptoms is due to one single misspelling, one nucleotide change can cause these devastating effects in one of the GRIN genes that encodes the subunits that make up a really important receptor called the NMDA receptor. So these individuals have really debilitating symptoms, including, as I mentioned before, intellectual disability, epilepsy, also problems in speech, vision, motor function, digestion, behavioral outbursts, and disrupted sleep. So every aspect of their life. Now, I learned about this because for many years I had been working with mice that had an induced mutation in the GRIN1 gene. So with targeted mutagenesis, we went and created a knockdown mutation in the GRIN1 gene. These mice have really low levels of GRIN1, and it causes them to have low levels of functional NMDA receptors. So we made these back when I was a graduate student, and I've been studying them ever since. And so we had the chance then to study what happens to the brain when NMDA receptors are not working. We looked at the dopamine system a lot, actually, and they do have hyperdopaminergia, more dopamine neuron firing all the time. They have deficits in all forms of learning and memory that we've tested to date, and they do exhibit spontaneous seizures. So even though we, studied these originally in the context of schizophrenia, as we learn more about GRIN disorder, we realize this is actually really nicely phenocopying what's seen for these children. So in the process of studying what happens to the brain, we did a lot of work with looking at molecular changes, either with transcriptomics or proteomics. And not just our lab, but other labs did this as well. And a common finding was an upregulation in the enzymes that feed from glycolysis into mitochondrial respiration. And at the same time, we knew that these neurons, a lot of neurons were firing more, cortical neurons, dopamine neurons. So there was just this state of neurons firing a lot. So neurons firing a lot means they need more energy. And so upregulating mitochondrial enzymes makes a lot of sense. But then we also saw this really strange observation, which was that the transporters that bring glucose, the major energy source for the brain, those transporters were downregulated. So this is a problem. If you need more energy and you're not bringing glucose in, that's a real problem. And so this diagram is just showing how does glucose get into the brain. It goes through these glucose transporters from the blood through the endothelial cells into astrocytes. Astrocytes undergo glycolysis and turn that glucose into lactate, and then the lactate gets brought into the neurons, and the lactate is actually what gets fed into mitochondria to really make the ATP that the neurons need. Okay, so we realized there was probably a bioenergetic deficit in these animals. There were other evidence that these glucose transporters weren't working. For example, we knew that 2-deoxyglucose uptake was down in these animals. So really problem there. So we tried to think, how can we help these mice? they're not getting glucose. They can't use glucose. But we know that they likely need energy. We know that people who are treated, who have glucose transporter deficiency, they're treated with a ketogenic diet because they can't use glucose, so they need a different source of energy. And that helps with their symptoms quite a bit. So we asked whether a ketogenic diet would help these mice, would provide the energy source that they're not getting from glucose. And you know, when you give a ketogenic diet, it changes a lot of things. So it increases energy production from mitochondria. It decreases inflammation. And it also, of course, is an alternate energy source. So we wondered whether just supplying this extra energy source, which is the ketone, beta hydroxybutyrate, whether that could do the same thing as a ketogenic diet. So we did these two things. We compared ketogenic diet with a control diet. And then the other thing that we did was we gave kind of the normal diet that mice normally get. And we just added beta hydroxybutyrate to what they're eating. So there are two different studies and they have different control diets. So we can't really directly compare them. We compare them with their control diet. So on the top, I'm showing you what we did with the ketogenic diet, where we started the ketogenic diet. Well, first we started it at 3 weeks, which is when the animals are weaned from their mom. But we found that they did not do very well. They didn't transition with the weaning. So we moved it back to 4 weeks of age, which is still quite young. We put them on this ketogenic diet. And this was something that you purchase. This is what most people buy when they study ketogenic diets in mice. So it's a very, very low carb diet. And it's also, a lot of the fat comes from Crisco, which is not a great nutritional source of fat. So I just need to emphasize that this is not the same ketogenic diet as what we give humans. Okay, so then we made sure that we were reaching ketosis by measuring their blood BHB. We did this 1 week after and then 9 weeks after, and then we looked at their behavior and then we did histology. So on the second study, we just kept them on the normal chow and then added hydroxybutyrate into the water that they were drinking. We did this at 3 weeks. We measured their blood BHB 1 week after, 9 weeks after, and did the same behavior and histology. So here's the first thing that we discovered was that these mice do not do well on this particular diet. Whereas we didn't lose any animals in, you know, that were wild type or that were the knockdown animals given the control diet, we lost about half of the animals. They died, you know, by the time we were going to test them. So what we take home from this is that these growing animals probably need more carbohydrate than what this diet was giving them, and that also this keto diet, where most of the fat comes from Crisco, is not supplying them the energy that they need. Okay, but then we still had animals that did survive, so we did our remaining studies on those surviving animals. So clearly there's a survivor bias in what we saw. Okay. In this, I'm showing you what happens to the blood ketones at 1 week and at 9 weeks. So we can see that in both the wild type animals and in the knockdown animals, we see a dramatic increase in their blood ketones. So they are in ketosis. And then this really diminishes after 9 weeks. And so what we think is happening there is that the body is actually using these ketones and that they're being absorbed better. And so the circulating levels are not as high. We also did the same thing with the BHB animals. We see increases in beta hydroxybutyrate, but they aren't to the same extent as what you see in ketosis. Nevertheless, we see the same trend where you see higher levels 1 week after treatment than you see 9 weeks after treatment. So we think that there are some compensations that happen to utilize these ketones that we're supplying. So what happens to their behaviors, which is what we were really most interested in? So this first behavior that I'm showing you is their locomotor activity when you put them in a new environment. So when you provide novelty, you get a surge of dopamine, and that increases their activity. And so in the black lines, you see wild type animals put in the new environment, and you can see that they travel a lot, especially in the beginning, and then they settle down and habituate. In contrast, the knockdown animals have really high levels of activity in the beginning, and they don't habituate. So the open circles that are in red show you the knockdown on the control diet, and then the closed triangles in red show you what happens with the ketogenic diet. So we're improving this dopamine mediated hyperactivity. We saw a very similar effect just by providing the beta hydroxybutyrate. So this was after 9 weeks of treatment with the beta hydroxybutyrate, but you can see that it significantly improves their hyperactivity, although it's not normalizing it. Another dopamine-related behavior is sensory motor gating, which is a measure of arousal, and it's kind of how high does the animal jump to a startling sound. And this can be attenuated if you precede that startling sound with a softer sound. So really this is measuring kind of what's their state of arousal and how well can they dampen that arousal state. And so this is complicated, and without a pointer, I am just going to have to verbally show you. On the left-hand side, we're looking at the effects of the ketogenic diet on these animals. And on the right-hand side, we're looking at the effects of the beta hydroxybutyrate. And so you don't see a lot of stars indicating substantial effects, but what I can say is that the knockdown animals in red have less of this sensory motor gating than wild type animals at these 3 different prepulses, particularly the lowest prepulse. And the ketogenic diet doesn't really improve that. On the other hand, on the right-hand side, you can see what beta hydroxybutyrate does. And we didn't see a significant effect. However, we do see an effect at these lower decibel frequencies, decibel intensities where you see the biggest deficit. So I interpret this that there is some improvement also in sensory motor gating with BHB, but not with the ketogenic diet. We looked at a lot of other cognitive behaviors that are impaired in the knockdown animals, but we really didn't see improvements with either the ketogenic or beta hydroxybutyrate in those. So it was really more the dopaminergic driven behaviors where we saw the biggest impact. Then we did this histology. And so one of the things that had been observed is that beta hydroxybutyrate improved myelination, and this was in a mouse model of Alzheimer's disease. So we were using the same protocol that this Alzheimer's study used, and we wondered whether we would see the same impact on myelination. Another reason why we were interested in myelination is that we knew these mice had volumetric deficits in white matter. So we were looking to see, first, what's the reason for these volumetric deficits? And second, could they be improved with either ketogenic diet or beta hydroxybutyrate? And so we did EM, electron microscopy, to look at the myelination in the corpus callosum. And we were measuring the amount of myelinated axons and whether we saw any of these abnormal patterns of myelination. And this would be, for example, some of the ruffling that you see where the myelin isn't tightly compacted around the axon. So what we saw was that there was no change in myelination, and there was actually an increase in the amount of abnormal myelination in both wild-type animals and in knockdown animals. So what we think is happening here is that this diet is not great to give to developing animals, at this really early age. And so, it's either the fact that they didn't get enough nutritional fats or there wasn't enough nutritional carbohydrate at this developing age. They didn't do so well. On the other hand, when we gave beta hydroxybutyrate, here with this diet comparison, we could actually see that myelination is reduced in the knockdown animals. So if you look on the right-hand side at those bar graphs, you can see that the untreated open circle knockdowns have fewer myelinated axons than their wild-type comparator. When you give the beta hydroxybutyrate, you see a level that is not significantly different from wild type, although the differences between the treatment and control are also not significantly different. So it's an improvement, but it didn't reach statistical significance. And we didn't see really a big improvement in the amount of abnormal myelination, but if anything, it is reduced. So that's an improvement over the ketogenic diet. So my concluding thoughts on this, I didn't show you a lot of the data that was the background, but we know that NMDA receptor dysfunction leads to increased neuron firing, which is energetically very expensive. There's a lot of upregulation of the TCA glycolytic enzymes to help meet that demand, but glucose transport is down. Some of the deficits may be partly ameliorated with a ketogenic diet or with beta hydroxy supplementation. Ketogenic diets require careful medical supervision. So I think that especially giving in a pediatric setting where kids are growing, they need a different kind of intervention than adults. The beta hydroxybutyrate supplements were well tolerated and improved some behaviors with modest improvements in myelination. So what we're doing going forward is to study the effects of beta hydroxybutyrate in patient variant mice who have the same misspellings that people have, also looking at things like their mitochondrial function, inflammatory readouts, and how BHB might impact seizures, which occur in the people and in the mice. So with that, I want to just thank the people who did this, including Phoebe, who is in the audience and is one of the scholars who's been funded by MITO2i. She's going to tell you more about the mitochondrial changes in these mice. And also Tatiana Lapina, who is a research associate in the lab, and they really did all of this work together. And also Rob McCollum-Smith, my collaborator, who helped with the glucose transport study and the proteomic study. Okay, with that, I can take any questions you have.
Thank you, Amy. That was really fascinating. So first of all, are there any questions in the audience to start with? Yes, Hannah.
Thank you very much. Should I use the microphone? Because I know that otherwise I cannot hear online.
No, I'm sorry.
Thank you very much. Thank you, Amy. And I think my curiosity is your thoughts on the quality of the diet, because as you really emphasized in the beginning, I think it is maybe give a little bit more of idea of why you think this possibly happened. And the second part of the question, I'm fascinating about the fact that you have a high relationship with the dopaminergic behaviors, which tied so well with what Mary Phillips was talking in the beginning. So just want to hear a little bit more your thoughts on the quality of the diet and the relationship with the dopamine. How could we study further and how we could be next to further understand potentially your models into the glutamate?
Thanks, Anna. So I learned a lot in this study and actually got involved with Bazuki kind of too late because this study started well before I started hearing from clinicians who were giving ketogenic diet to people. And so Crisco is really good for keeping fats, it's a room temperature stable fat, and it's great for putting in mouse chow where you don't have refrigerated food that you then give to the animals. So it's a lazier way to feed animals. But I think it would be better to have refrigerated food that was closer to what we actually are interested in studying. And animals normally eat seeds and nuts, and mice they love peanut butter, they love nuts and sunflower seeds and things like this. So that's probably more what they should be eating than Crisco. So if I were to do it again, I would look for something that was more naturalistic. And I think there have been some studies where they use seed diet in mice. So I think that would be the best thing. Okay, I can't remember now the second question. Oh, right. Yes. So I was so excited when you started talking about dopamine, and I wish I had known this when we wrote up the paper. So the paper's in revision now, but this was really exciting to me to see that connection. And yes, so there's now a lot that we can do to study the dopamine system in these animals. We know that their dopamine system is dysregulated. So now we can go and look and see all of these different ways that you can study dopamine in mice. We could go and look to see whether these interventions, particularly the BHB, which is so much easier, actually change. And it kind of, mechanistically narrows down all of the different changes that happen. I forgot to say, like your microbiome changes, and, there's a lot of things that could possibly explain it. So, yeah.
So I have a question if there isn't, but first of all, is there another question in the audience? See, yeah, okay, well, I'll go last then. I think Ruth, I saw you first and then, yeah.
I don't know an enormous amount about the ketogenic diet. I've read up on it, but not from a scientist perspective. So I work in the endocannabinoid system and bioactive lipids, and these are really important signaling molecules, and their levels are completely affected by diet. So is there data you were talking about that was a kind of substandard diet for the mice, but just in terms of the different omega components of a ketogenic diet and how patients respond, and because I think so much of the analysis is done based on ketosis and ketone bodies, but is the actual lipid composition really really something that's key to the effect?
Right. So I know that that's a great question. I know that these mice are really vulnerable to omega-3 deficient diet. So if you take out all the omega-3 from their diet, then the males die in the same pattern. So I think that it is likely this deficiency in fat that is part of the reason why we see this actually. Mary, you may have some thoughts on that as well.
Well, I was going to ask something related to that, but I think there were some other questions, and I feel guilty about taking the scene. So I think, yeah, I think you had a question, yes, and then. Kirk.
Many people, when they use a ketogenic diet, achieving a certain level of beta hydroxybutyrate is their goal.
Yeah.
So are you surprised that where you actually gave BHB that, the results were not what you expected? So you're talking about deficiencies of other constituents in the diet. Is that right?
I was surprised to see them, yes, not flourishing. And actually, so some of these children are so ill that they need a G tube, a gastric tube for feeding. So in that case, it's very easy to give them a ketogenic diet. And there have been reports that parents, that they start really gaining weight. All of these kids are underweight, as you might expect, and they gain weight, they do better, they have fewer seizures. Cognitively, they seem better. These are anecdotal. I don't think there's really been a published study on this. So yes, I would have expected that the mice would get better. The ones that survived did gain weight. So they did gain weight, but then we have a survivor bias that we didn't see those that dropped out, but they didn't gain more weight than, the ones fed the control diet. So yeah, I was surprised, and the results are what they are. So I think we just have to interpret them with this knowledge and not over interpret them,
That'd be quick, thanks. I can sympathize with mixing Crisco and powder and butter and stuff on a daily basis to feed animals. So my one comment was around the macronutrient ratio. So 6:1 is not something you'd ever see clinically. I know at Sick Kids, when I worked in the ketogenic diet program there, 4:1 was seen as sort of the most restrictive, rigorous form of ketogenic diet. And in a lot of the clinical trials that we're involved in, we're seeing ratios of 1.5 to 2:1. I think one of the issues is whether you can get, a balanced diet with 6:1. If you're actually getting enough protein, would be my concern. I think carbohydrate as well, but and the
Right.
impacts of that. So I think it's something to consider.
Right. So the diet that we're using is the one that is published for mice for doing a ketogenic diet. So this wasn't something that we formulated. We're like, well, let's do what everyone is using, so that we can compare our results with everyone who does ketogenic diet in mice. So if you look to find what is a commercially available ketogenic diet in mice, this is what you'll get. But I agree, it's very extreme. And so this is where, if I had been in the kind of metabolic mind arena, I would have probably started with a different diet. Yeah, it is extreme.
There's one question online, but can I ask my question? I've been waiting, but I've been fascinated to hear everybody else's questions which relate to mine. I mean, I have several questions, but I'll just limit it to one. Do you think there's a critical developmental period here? Because you were dealing with sort of a specific age group. And I'm just thinking, in humans, obviously myelination occurs probably up until sort of mid-30s. But we know that in the first 2 years of life, and I also study infant development as well, that, these networks of myelination really, really, increases a lot. So I'm just wondering, maybe the keto diet, that there's a critical period when it has to happen, before which is too soon, after which may not have great effects. And maybe you just got your animals too soon. I don't know. I just, looking at your myelination pictures, it's quite profound.
Yeah. So all of those were scored blind, so there was one person who took the pictures, a second person who assigned them, and then a third person who scored them. And so, we saw this ruffling and we just assumed, well, these are the mutants that aren't treated, because we knew that there were these MRI changes. And so then when we found out, oh my gosh, these are wild types treated with this diet, that was surprising. And yeah, the question of the critical period is a great one. And I think the answer is yes and no, because I think absolutely critical periods are important. And whenever, whenever you can intervene to help a brain, the better, right? The sooner, the better. On the other hand, we've done a lot of work with these mice where we do kind of adult rescue, genetic rescue, and we see tremendous improvements. So I think there's like this plasticity of the brain, that this ability to, if you give it what it needs, it can, it can reverse a lot of problems. Maybe not as well as if you give it early, but so yes and no. And it's a great question. I mean, that idea of when to intervene is so important for any neurodevelopmental.
And I think, as Kirk and others were saying, the actual constituents of the diet presumably are critical as well, and they may vary at different developmental periods as
Right.
well. You know, what's needed. Ketosis may be different in a child than for an adult. You know.
Right.
I believe, but thank you very much. I think there's one question online. Anna.
Yes, I will be the online voice. So Peter Carlin is asking you, has BHB been tried as a treatment for anti-NMDA encephalitis, a devastating disease?
Okay, yes. So anti-NMDA and encephalitis is when people make autoantibodies against their own NMDA receptors, and it kind of starts looking like psychosis and then degrades into seizures and then coma and death if untreated. I do not believe that it has been tried. The main so now when people present with this, they're tested for, do they have antibodies against this receptor, and they're given immunosuppressive treatments, and that works really well. I don't know whether there are some people who never recover back to normal, and I don't know whether BHB could be, a treatment for that. So that's a really great thought, and I will go look at the literature, but I kind of have my ears perked for those two things. I haven't seen anything on that.
Sorry, can I just ask one question related to that question? Can you just say very briefly, what's the link between NMDA receptor dysfunction and the glucose transport?
Aha, yes. Okay, so this is one of my favorite observations, because it got me thinking about the NMDA receptor in a very different way. So there are some studies showing that when you apply NMDA receptor antagonists to cells like astrocytes, oligodendrocytes, neurons, culture, you see a decrease in the transcript, the GLUT1 transcript, or GLUT3 transcript for neurons. So I was just very curious. It looks like the NMDA receptor activity regulates the transcription. Why is this? So Chris Anderson has published some really nice studies and reviews talking about NMDA receptors on other cells, like endothelial cells, astrocytes, and their role in sensing neuron activity and responding accordingly. So glutamate it is released whenever neurons fire. And so other cells can sense it, and they use the NMDA receptor as the way to sense neuron activity and then respond accordingly by upregulating the transporter. So it's almost like the neurons are shouting, feed me, and the astrocytes and the endothelial cells can't hear. And so they think everything's fine. And, that is my working hypothesis for what's going on. Yeah.
Thank you very much indeed. I think we're at the end of the session, I believe. So let's just give a big round of applause to Dr. Ramsey. Thank you.